Hey everyone, so the other part of this is going to be taking a look at the story of genetics. Basically, we have a lot of evidence from the fossil record, but it's incomplete, of course. But what kind of story would our genomes tell? Well, in order to get into that, we need to first do a little exercise here. I mean, there's of course a few striking things about the fossil record, clear patterns and changes in morphology. There various intermediate transitions or major transitions, if you would like to call them that. But it turns out there's an even more precise way to go ahead and test this hypothesis, if you will, of common descent with modification over time. And that has to do with how manuscripts, the copying of manuscripts. So let's suppose you have an original manuscript and then it's copied twice, and both of them have their own unique typo. So now let's say you find a third printing that just so happens to share the exact same typo on page 14, but then has its own unique typo on page 8. Now, an obvious question is well, was it copied from the original manuscript, copied from the first printing, or copied from the second printing? Let's say there were only three possibilities in total. Well, the most parsimonious explanation is that it was copied from the first printing, and that typo on page 14 was propagated into the third printing. Now you could do this with more manuscripts, and essentially you have, now let's say you have a fourth printing, but that one shares the typo on page 14, but does not share the typo on page 8. It has its own unique typo on page 3, so you could ask, the fourth printing, was it copied from the first, second, or third printings, or the original manuscript? Now, it could have been copied from the original manuscript, but they just so happened to make the own unique typo of the first printing and a separate one on page 3. It could also technically be copied from the second printing, but instead it undid the typo on page 23, did the typo on page 14, and then had a new typo on page 3. And you can see how these types of explanations become more and more convoluted. Now, you could never really know for sure in this if you were only looking at a few various typos, but if you start to look at hundreds and hundreds and thousands of unique variants, you would be able to piece this together really, really well. So here's an example if you had a sixth printing. So let's say you have a sixth printing here that has the same typo as page 14 and 3, but a unique one, never seen before in any printing on page 5. Where would you put it? Here, what you could basically, it's pretty obvious. You would put it. Let's see if I can. Doesn't seem to be drawing, or maybe it is. I can't see it though. But anyways. Which of these is most likely? All of these could be true. The sixth printing could be a separately authored book that's not a copy of the original manuscript of which the 5 printings are copies. That could be the case. But it just so happens to have all of the same words as the original one and all of the same shared typos as the various printings. It could be a direct copy of the original, but the editor could have independently made 2 different typos on different pages and a new one. It could be a direct copy of the first one, but then share the typo on page 3 and then it's a new one, or it could be a direct copy of the fourth one. Um, and so the most parsimonious explanation is, of course, the bottom one. But all 4 of those are possibilities, and you could even have more exotic ones as well, where it could be you could have the second printing, and it could undo that typo and then gain the other typos independently. That could be true, but this is the most likely solution. So you can have that, of course, with mutations. Same type of thing. Basically, at the point of separation, you just have some population group that's splitting, and the two basically branches would share. They would have identical books or genomes, and as they go their own separate ways, they accumulate their own mutations. We see this on small scales, say, for example, within the human population today. You can have unique variants, and that's how they do the 23andMe or different DNA ancestry types of things, where you basically have unique variants, the basically unique typos that were shared and accumulated in population groups that once split. And so you could, of course, have an accumulation of mutations, and you could figure out where and exactly how different population groups are related to other ones. And we'll look at that in quite a bit more detail when we look at human beings. But here's just an example. You could have, I'm not going to go through this, I'm just going to skip over it, so on and so forth. And it turns out you could get even more specifically and start to look at individual base pairs. And so let's say you had this little snippet here of, well, that's about 9 amino acids, I think, each pair of 3, a little more than that. And so here are 3 different species of fruit fly. And so you could ask, well, how were these 3 related to one another? Well, you could put together some kind of tree like this, where you could have the Yakuba, where basically you have some kind of common ancestral species here, and then it didn't have any changes. But then the common ancestor to simulins and Cicalia could have had a point mutation at point 4 in this particular gene, so on and so forth. And then they both had no further changes as the population groups split from there. And of course, an interesting question is, how could you tell what the original sequence is? Because it's possible that the original sequence could have simply contained an A at position 4, and then it was D. Yakuba that had that particular mutation. That's something we're going to come back to. It's a really good question. But at the same time, you could piece together that at least simulins and Cicalia are most likely more closely related than to each other than they are to D. Yakuba. Um, and so an interesting thing about this is, of course, there's some wiggle room in randomness, which is basically each 3-letter sequence codes for a particular amino acid. So if you want to go ahead and follow along, you see most of the amino acids are here, are the same, except that simple base pair change changed the actual amino acid sequence. And there's just a list of them. But one of the interesting things is this, that you can have multiple sequences can produce the same amino acids. So basically, there are many ways to write this sentence and arrive at the same meaning. So let's say, for example, you needed to have this particular sequence of amino acids. So you can get a really interesting effect, and this is a little more detail, an example of the wiggle room you can have in randomness. If you need the same sentence, the same amino acid sequence for the same function, you could write the individual base pairs quite differently, because you can have multiple combinations of base pairs that code for the same amino acid. So here's just a hypothetical example where only 14 of the 27 monomers actually match, but yet this species would have the same exact function. So that's a really important thing to keep in mind. And so that's just an example of all of the possible differences. And the point is, and this is going to be really important, is that genes match much more than they need to. So here's an example of a 4th species of fruit fly. I'm just going to skip over that for now. But this is really, really important. So let's say there are 5 species here. And there's no need for these sequences to be so similar. There are 53 million ways to code for the exact same amino acid sequence in these 5 species. But you see there's a lot of similarity between them. The bottom 2 have some more shared mutations, so on and so forth. If I go ahead and label them, you see that there's humans, chimpanzees, gorillas, orangutans, and dogs and wolves. And this particular segment works for a gene for smelling, and it turns out the dog and wolf sequence works quite well to perform the function of this gene in particular. And so the point of this is that this particular sequence matches much more than it needs to. This sequence could be, you could have the same amino acid sequence and change the base pairs in 53 million different ways. So for example, you could write the Homo sapiens one. So let's say Homo sapiens are not related to any of these other species listed here. Well, they could be completely different. You could have the Homo sapien sequence be pretty much anything, one of the other 53 million possible ways. I'm not sure statistically how much you could get the monomers not to match, but they match way more than they need to. And of course, if you do this, and we're going to talk more about this specific number, what does this number mean? If you compare the actual difference in DNA monomers, you get that a similarity of somewhere around 99% or a little bit more than that. So that's a very, very impressive amount of similarity that matches much, much, much more than it needs to, even for similar types of functions. There are various things. Humans, for example, crawl when they're babies, and so do chimpanzees. They crawl when they're babies in a fairly similar way. And I'll show a picture of that in a little bit. So that's just an example of a trait that's in common. Or, for example, we both have immune suppressing genes that happens in the mother's womb whenever she's pregnant. And they actually happen to function in the same way. We'll look at that a little bit later. But this is something that's interesting, a data point, if you will, that basically base pairs match way more than they need to have the same function. There's absolutely no reason why the Homo sapiens sequence could have been completely different for the same function. Here's another example from the gulo enzyme. So it's basically required for the synthesis of vitamin C in mammals. Here's an example of 3 functioning vitamin Cs. This gene is fairly highly conserved between mammals, and the reason is because if this particular gene, it's under selection pressure for most mammals. If this particular gene goes bad, animals will get some kind of equivalent of scurvy. Obviously, if they can't produce their own vitamin C, there's a malfunctioning gene they have that most likely will not be propagated throughout the populations. Now, of course, the interesting thing is that humans, we can't produce vitamin C on our own. It turns out we're missing the last part of this pathway. So we have the whole pathway except for this last little part. And it just so happens that one of the reasons why this is non-functional in humans is because we've essentially had a base pair deletion. That's it. Just the removal of one DNA monomer highlighted in that yellow in the primate sequence. This deletion is identical in humans, chimpanzees, and orangutans. Basically, it's a shared typo. So to go back in that analogy, it is possible that humans had a functioning gene and they just so happen to get exactly the same typo as chimpanzees and orangutans. That's possible. It's always possible for that to occur. If we were just looking at one gene in particular, you couldn't really say for sure. I mean, it would be fairly and highly unusual for something like that to happen, to have a base pair deletion. Maybe changing from an A to a T or something like that might not be so rare, or something like that. But this one in particular is really interesting. So if you were going to make a phylogenetic tree on this, this is how you would do this, where you would put the loss of gulo happening since it happened in the exact same place. There's many ways you could have a non-functioning gulo gene, but it's very significant that it happens in very similar spot in humans, chimpanzees, and orangutans. So the conclusion is that it would be lost in an ancestral population to those 3 different species. So that's basically how, if you were looking at this in the most parsimonious explanation, you would conclude that this would be the case in particular. So there's a lot of different genes that you could look at. You could ask, well, I don't know, this is really hypothetical, so I don't even necessarily like this question, but it's an interesting one. What would it look like for a common designer to have made a functioning gulo gene in all species? Well, we could start out with completely different sequences, and then just over time humans lost ours because there wasn't high selection pressure in various early hominid groups that lived in regions that had a lot of vitamin C. So we essentially lost there was no selection pressure on that particular gene because we were getting it from food sources. Or another thing is that we would all start out with identical gulo genes, but then random mutations would accumulate over time, and there should be no pattern to these particular mutations, but there are. And it turns out that different genes under different selection pressure, you can take a look at it. And you could find when different organisms branch from one another. So that's what's showed on this. Here's cytochrome C that's had a lot more selection pressure on it. And then there's some others up here as well that you could go ahead and ask and compare. There are a lot of genes that are conserved, and you can compare the accumulation of mutations in different organisms, and you find a very consistent result. The consistent result shown here on the left here is the differences in various genes. And you just see an accumulation of random mutations in particular genes in different population groups. And we'll again look at that in a little more detail. So a really interesting thing to pay attention to are what are called pseudogenes. They're basically the remnants of once functional genes. Um, they can, of course, have functions, but very few of them actually do. There's a very large number of such pseudogenes in the human genome. And whether or not they actually have function, that's not exactly the point of them. But some of them do, but very few of them do. But anyways, they're pretty easy to spot. So for example, you could just simply, if you follow that pseudogene, you could do the same type of thing. I'm not going to go through this. So you could just ask, hey, which species have pseudogene X and Y? And you can piece together a phylogenetic tree based upon which particular pseudogenes they have. And you could say, well, you could even have pseudogenes that are unique to particular lineages. So when did that pseudogene, when did that mutation occur? Well, it occurred sometime after, in this particular case, species C split from the other two. So this is a really interesting thing. This is the power of genetics, and this is a really, really important point. I mean, if life really does have this kind of backstory, this common ancestry, you could really trace the history of life species by species. I mean, of course, there's been other non-genetic ways. So, for example, you had paleontology or comparative anatomy, and so on and so forth. Those have been worked out really well before DNA sequencing came to be. And if the DNA data had suggested completely different lineages, there would be good reason to doubt this common descent hypothesis. But that's not the case. I mean, essentially, in the scientific community, there's no longer any doubt that common descent is the best hypothesis for living things. And the genetic story is really, really impressive. So here's just an example again of the pseudogene thing, just kind of jumping back to that. And it turns out you can trace this and ask, well, what about pseudogenes in humans, chimpanzees, gorillas, and orangutans? And it just turns out, if you go ahead and look at this, we have 15 unique pseudogenes. This is only looking at 47 of them. We have over thousands of these pseudogenes that you can construct much more elaborate diagrams. But they give us the same result, that humans are most closely related to chimpanzees. So it just so happens that humans have 15 unique pseudogenes in this little representative sample of 47. Chimpanzees have 4. And it turns out 3 are shared by humans and chimpanzees. That basically we would have a lot of confidence that those 3 are shared by humans and chimpanzees because of a similar base pair deactivation for different genes. Or you can have a frameshift mutation or deletion, so on and so forth. And then it just so happens that we also share 3 with gorillas as well. So gorillas have 5 of their own, but gorillas, humans, and chimpanzees share 3 in particular. And then we also share 6 more with orangutans. So a common ancestor to humans, chimpanzees, gorillas, and orangutans would actually share 6 pseudogenes. And you could basically build this more and more. It turns out there are pseudogenes galore. So for example, switching away from human beings a little bit, there are genes for teeth in birds, turtles, and toothless mammals. Here's just an example of some pseudogenized genes and pangolins. This is really, really interesting because we do have ancient bird-like fossils with teeth, ancient turtles with teeth, and primitive mammals have teeth. All of them do. So that would indicate that if there are any mammals without teeth, they once had them and since lost them. So it's really interesting. We had fossil evidence, even before looking at teeth pseudogenes. And so it turns out that we can find various deactivating mutations. So here's just an example of a large base pair insertion. You can see that region in the blue in the graph on the right, and then another one there in a base pair deletion. But then outside of those sections, you find the identical nucleotide sequence. And so what would that let you know is that this particular gene has been deactivated, but it once had an active functioning gene. Another interesting thing you could look at is related to insects. This is an interesting hypothesis that one can ask because early mammals, if we ask what was their diet like, well, they had really small teeth with sharp pointy ends, characteristic of modern insect-eating mammals. If you compare this with mammals that arose, of course, after the end of the dinosaurs, Well, of course, they had much larger body sizes and a much wider type of diet. And so why did mammals change? That's an interesting question. Obviously, you could have an exploration of new ecosystems. You didn't have the selection pressure to stay small that you would have had, of course, from various predatory dinosaurs before this point. And they were basically constrained by dominant land animals. But an interesting question is that if mammals once ate insects, is there evidence in the genomes of mammals that we once did? And it turns out, yes, there are 5 different genes used for digesting insect chitin. Modern mammals that have all 5 is functional, basically consume mostly bugs. In modern day meat and plant eaters, every single species has pseudogene remnants of 1 or more chitinase genes. So that means sloths, elephants, manatees, fruit bats, horses, rhinos, camels, rabbits, tigers, polar bears, walruses, dolphins, oh my, have remnants of genes that were once used to digest the prey of their ancestors, insects. Humans also have 3 chitinase pseudogenes and a functional chitinase gene. 2 of the pseudogenes, interestingly enough, share the same deactivating mutations with monkeys and apes, and the third one, we just so happen to share the deactivating mutation only with apes. And so that's really, really interesting. And so, interestingly enough as well, many of the earliest primate fossils also appear to have been primarily, or at least had a higher insect type of diet that you could tell based upon the jaw structure and the teeth as well. But as plants and meat became more prominent in such diets, the insect digesting genes were likely less useful. You also have remnants of third eyes on various lizards. There was already evidence from the fossil record, in particular, a sequence of fossils from primitive reptiles to the earliest turtles. And it just so happens that there was a pseudogene for this. Also, why don't cats like sugar? Well, it turns out cats and a number of other closely related carnivorous mammals have pseudogenized versions of the genes that bind to sweet-tasting molecules. So at one point in the past, why would cats have a large portion of this particular gene? Well, at one point in the past, they would have been able to taste sweet things, but they've had a deactivating mutation that just so happened to share with their most closely related common ancestors. And an interesting thing you can look at is also synteny. So that is not just comparing a little region of a gene, but it's comparing where is this particular gene relative to other genes along the genome. And so here's an example of 2 species where you have something called synteny, literally means held together, where genes appear in the same order on chromosomes. So if you have 2 closely related species, you would find gene A, and to the right of gene A is gene B, and then gene C and gene D. So an interesting thing, of course, is this particular study or example from looking at the vitagelin gene. So chickens, they have 3 particular vitagelin genes, vitagelin 1, vitagelin 2, and vitagelin 3. And then there are some neighboring genes that aren't involved in making egg yolk. They just so happen to be the closest neighbors. There's nothing in particular about them or special about them relevant to making eggs. They're just the closest neighbors. So interestingly enough, it turns out that humans have functional genes, these 3 genes in particular. And so, and the spacing in between them is very similar, even without looking to see, do we have actually remnants of the vitagelin genes within our genome? And that's an interesting question. And it turns out that within the functional genes, so that would be ELDT1, SSX2IP, and CTBS, that region is fairly conserved. So the black bars represent shared synteny. And you see if you go look in the vitagelin regions, especially in vitagelin 1, but there are aligned sequences for vitagelin 2 and vitagelin 3. And so this is a really interesting result. What does this tell us? Well, it's an interesting question as to why you would have matches in the base pair sequence to the right of these particular genes that are conserved in humans and chickens. It's a very interesting thing. Another interesting thing you could look at is what about a mammal that has a more recent ancestor that laid eggs. So for example, if you go within the common ancestry model and ask, when did humans, at least our ancestors, last lay eggs? It was a while ago. Um, but an opossum is fairly closely related to other egg-laying mammals like the platypus. No, platypus. Yeah. And so interestingly enough, they have a much more higher conserved region in the um, yolk region in Vitagelin 1, 2, and 3. So it's a really, really interesting thing um, there. And ERVs are another thing that we'll just take a look at, and we'll end with ERVs here. This is a lot of stuff, and I'm trying to blaze through it, probably not doing justice. Let me know if you have any questions on any of this in particular or various references. Most of them I think you should be able to find, but basically this is really interesting. This is what ERVs are like. Basically, ERVs, in order for these retroviruses to replicate, I'm looking at the bottom bullet point here, basically they insert, it has to insert its genome into our genome that then gets replicated by our material. And it's pretty random how this happens. And it just so happens if it happens in a, an egg or a sperm cell, then this viral insertion can be passed on to offspring. And this is would basically be called an endogenous retrovirus. And so the question is, are these insertions really random? If they are, and they just so happen to be in the same place, that is incredibly, incredibly remarkable or unlikely that they would insert into different lineages in the same spot. It'd be like having two Oxford English dictionaries, and you give one person a dictionary and another one, and they go into separate rooms and pick 100 random words. And then they come back, and if you found, it would be really, really remarkable if you, what would be the odds of finding all 100 words, or nearly all 100 words from two random searches matching up? Extremely impossible. And so that's a really interesting thing. And it just so happens that we have about 200,000 ERVs in our genome. And it just so happens that out of these 200,000 ERVs, they're shared by any siblings you might have, extended family, and pretty much all other humans alive on the planet. There are some unique ones that you might have, but by and large, this number is close to 200,000 shared ERVs amongst humans. And so this is a really interesting thing. You can compare this between species as well. So if you do this, here's an example on the left here. This is basically how many ERVs do we have. That's number of copies times 1,000. If you add those up, it's close to 200,000 ERVs. And on the right, this is comparing how many are unique to chimpanzees and humans. Oh my goodness, my child's cat is meowing, a pretend one. So this is the number of lineage-specific insertions. And so out of those 200,000, how many are unique just to humans that we have that chimpanzees don't? Well, 82. How about for chimpanzees that they have that humans don't? About 300. So out of the 200,000, we only differ by a tiny, tiny percentage. So this is a really, really interesting thing that we share basically all of these ERVs with chimpanzees. The question is, how could that possibly be the case? There are really a very small number that are unique to us and that are unique to chimpanzees. And so how did we get all of these shared ERVs? Well, there may be a couple of things. I mean, obviously, we could do a simple calculation like this. We could say, did they all insert independently, or are they passed down through common descent? If we want to reject common descent, how can we explain this? Well, if they did insert randomly, the probability would be something like this. The odds of ending up in a particular spot are about 1 in the length of the genome, or about 3 billion base pairs. And then the odds that all 200,000 just so happen to insert in the same spot, it'd be 1 over 3 billion to the 200,000th power. That is a stupidly large number, or small number. So that definitely didn't happen. So what does that tell us then? Well, that tells us, well, maybe the odds are different. Maybe there's some kind of way to explain this ERV evidence for common ancestry in a different kind of way. The odds are different. Here's an example. Can ERVs have site preference? So here's a nice review paper. It's called Molecular Mechanisms of Retroviral Integration Site Selection. Basically what these researchers were looking at, they were hoping that you could actually have site preference in ERVs. This would be huge in gene therapy and cancer treatment. or treatment for other genetically inherited diseases. CRISPR kind of does something similar with a site preference, um, and that's one of the limitations and challenges to it right now. We'll look at that another time. But really interesting thing is that most of the classes, when we look at this, I mean, this would be huge, but it doesn't actually work that way. Most ERVs don't show much site preference at all. There was one kind in particular that had half of their insertions within 2% of the human genome. But basically, reviews like this is that we've looked and we've asked, and the thing is that these insertions did not occur independently. It's not as if they have such high site preference that could possibly end up with 200,000 of these ERVs in the same locations across the whole genome. Another interesting hypothesis is another way to ask that is, well, obviously, while the vast majority of these would be inherited, they obviously had to have infected the genome at some point. So here's an interesting study. This is a 2005 paper that was looking at 287 retroviral sites. This is looking at some retroviral sites in chimpanzees, gorillas, baboons, and macaques. And so basically, it was looking at a particular region of ERVs, and they found that of the 299 they looked at, 275 of them occurred at non-orthologous locations. And it turns out that they weren't able to tell if the last little bit occurred at orthologous locations. Basically, an orthologous location would be that they occur at the same spot within the genome. A non-orthologous one would be that they don't. And so this is a really interesting thing. This is looking at 299 particular ERVs, and they found that basically none of them occurred at the same spot in the genome. And so this is what it would look like if ERVs were the result of a separate bombardment. So this is what it would look like if ERVs did not get in the spot that they were through common ancestry, but instead basically less than 5% at best could be in orthologous locations. That's basically the power of what this particular result shows. So basically, we can identify how ERVs got into different lineages. If they're in non-orthologous locations, it was through some kind of separate species bombardment. So, for example, these different primates, they were independently infected by different ERVs, and the ERVs in their genomes didn't occur through common ancestry. Now, they share a lot of common ERVs. These species in particular share a good, 200,000 ERVs, and they just have a few small differences. But this is just an example of looking at the differences in ERVs, and you can tell whenever ERVs are shared versus bombarded. So basically, this is a really interesting thing that we're going to come back to, but essentially you're filled with a long history. And again, we're asking this question. With nearly all ERVs in the same locations, how did this occur? Well, essentially, we know they didn't occur as independent insertions. At best, you could have less than 5% in homologous locations. It's likely much lower, this percentage. So essentially, we can reject the hypothesis that our shared ERVs with chimpanzees were obtained via separate bombardment. And the most likely explanation is, of course, common descent. The other explanation is they just so happen to be put there in a way that matches common descent. So, for example, a higher percentage of ERVs were put in humans and chimpanzees, and then a lower percentage of shared ERVs were put in humans and gorillas, and a lower percentage than that were put in humans and orangutans, and so on and so forth. And so, if that's the hypothesis, I mean, perhaps I've heard this argument here that they were just put there for functionality, and say that, well, yeah, that's the case. But it just so happens that these retroviral snippets have a lot of function. They do a lot of things. Some of them do. We're going to look at some that have been repurposed in our genomes and actually help with things like childbirth that I hinted at before. But they're essentially no wholly functional ERVs. really just a fraction of these original ERVs that have been repurposed for something else. And if this is one's hypothesis, it should be so easy to go and do this, but we can't. These ERVs are mostly not functional, so they weren't put there for some kind of function. Really, there's no way to explain how these ERVs got here apart from common ancestry, and that's why in the scientific community, things like this are pretty much very well established. There's no question as to whether or not common ancestry did or did not occur. It's pretty much a fact. So what are some ways to deal with this? We'll look at that next week, or in the next lecture, and we'll also look at some more things regarding humans in particular coming up.